Welcome to the Performance Around the Clock podcast. I am Professor Sachin Panda, a scientist in the field of circadian rhythm. In this podcast, I will talk to experts in many different fields about science, health, and human performance. You'll also find out why circadian rhythms is my favorite topic. I hope you enjoy the podcast and thank you for listening. Welcome to another episode of Performance Around the Clock, and we are recording from Lubeck, Germany, where I'm attending European Biological Rhythm Society Congress with Professor Rae Silver. Professor Rae Silver is a professor at the Columbia University, and she has a long history of very fundamental research in circadian rhythm. Welcome, Professor Rae Silver.
Thank you, Sachin. Thank you. So pleased to be here.
Yeah, many circadian researchers kind of started somewhere else, and then slowly they were curious about circadian rhythm and got into the rhythm field and made some big discoveries like you did. So how did you get into circadian rhythm? Because your work is really foundational in finding out the part of the characterizing, figuring out the function of really the master clock that's in everybody's brain. So how did you get to that?
Well, my first love of research started with birds. And what captivated me and made me so excited was when you look at the parental behavior of doves and pigeons, the entire family of doves and pigeons, the male takes care of the eggs and the babies from 10 o'clock to 4 o'clock.
Huh.
Isn't that amazing? And the female takes care the rest of the time. So basically they split the daylight hours. And I thought, how do they know what time it is? And so I did this huge number of experiments trying to figure out what are they timing. And then the other thing I tried to figure out is, how do they know whose turn it is? And then I tried to figure out, why are they doing this in the first place? Is it an obligation somehow? Or is it a privilege somehow? And being able to answer those kinds of questions just fascinated me.
But it still boggles my mind. Why were you watching dogs and who is taking care of the eggs? Because, in my backyard, I have a bird feeder and a bunch of dogs come, but I don't know where their nests are.
Aha. Well, that's a whole story. I was asked to join the laboratory of Danny Lehrman, and he was a very famous endocrinologist and one of the first people who joined neuroendocrinology. And he had as visitors all the ethologists in the world, all the people who studied birds. And he was interested in how behavior developed. So in his years, people explained behavior, it's instinctive. And he argued that instinct doesn't explain anything. You have to know how the behavior is stimulated and sustained. And so he studied how hormones affect parental behavior. That was his research area. And the part that I became interested in was, well, how do the sexes know what time it is? I knew which hormones affect parental behavior, but I didn't know how the male and the female could share that responsibility. And the answer is really quite fantastic.
Yeah.
So if you give if you put two males together, they just fight. They destroy the eggs. They would kill the young. They can't do it. If you give two females a nest and eggs, then they sit on it, they take care of it for a long, long time, well beyond the time of hatching. But there's no timing. They just sit. But if you give a male-female sex hormones, then they sit in sex-typical pattern. So the hormones are somehow telling them whose turn it is. The hormones are affecting time. And I thought that was a great problem.
So, but the dogs, they don't reproduce around the year, only in springtime. Or am I incorrect?
Well, like most animals, they produce most of their young in the springtime. But if you see them around the city in the fall, they will still be courting. Sometimes they lay eggs, but they won't hatch; they'll just die. So yeah, they're seasonally reproductive.
So that means your research was also initially seasonal.
Was also what?
Seasonal.
Oh no,
No. No,
we had them in the laboratory.
Okay.
They were in the laboratory.
So let's go back to that. So the dad takes care from 10 to 4.
Yep.
And then the mom has to take care for the rest of the day and night.
Yep.
Ah, that's not right.
I even have a 20-minute movie on this. You might think it's not right, but in fact, the only hours that they run around and do things, fly around and do things, is in the daylight. So in the night, they're just sitting still anyhow. They're sharing the daylight hours.
Oh, okay. And also these are birds. So there is so they just sit on the egg, and maybe the mom sleeps over the egg and until the egg hatches.
Right.
After the egg hatches, is there sharing of responsibility?
Absolutely, yes. The male takes care of the young from still same time, from about 10 until about 4.
I did not know.
And this is true for all members of the pigeon and dove family. So you can see this near you. If you see a bird and maybe put a little bit of vegetable dye, you'll see. Or sometimes the pair doesn't look the same. And you can tell who's who.
Yeah. And then if you just put the sex hormone, then that will trigger that behavior.
Right. Right.
That's exciting.
It is. And Jürgen Aschoff loved that work. We used to chat about it all the time.
Do other animals also do this kind of sharing of responsibility?
They do, but it's not temporally organized in that way. So, penguins will sit for long, long bouts and then trade places. So I don't know of any other animals that share temporally like that.
Yeah. So that was your PhD thesis, or?
Yep.
So then how did you get interested in?
So once I knew that the sex hormones did that, I was trying to figure out what the site of action could be. Okay. And at that time, the suprachiasmatic nucleus had just been discovered. And so I started looking for the suprachiasmatic nucleus in birds. And it turned out that the bird organization does not seem to be the same. So we did track tracing from the eye, which is the classical method, and the traces did not end up at the place that was identified as suprachiasmatic in avian brain. It ended up somewhere further laterally. So the avian model wasn't a good one for understanding effects of hormones on timing. So that's why I switched to hamsters that were a much better model. They had very precise rhythms, and we knew exactly where the clock was. And so I did a lot of work looking at where do sex hormones act on the clock. Of course, they don't have that beautiful timing behavior, but so it goes.
So those listeners who don't know what is suprachiasmatic nucleus, as many of you might know, or maybe you can describe why it is called suprachiasmatic nucleus.
Sure, it's very easy. So the eyes have tracts that cross between the right and left side, and the point where they cross is the chiasm and the suprachiasmatic nucleus in most species, all mammals that have been examined for sure. The suprachiasmatic nucleus sits on top of the chiasm, so suprachiasm.
Yeah, but that suprachiasmatic nucleus, although it sits on top of the chiasm, it's at the bottom of the hypothalamus.
Yes. Yes.
Of the brain.
Right.
And it's very well defined, very small region of the brain. Contains what, 10,000, 20,000 neurons?
In rat and mouse, it's about 10,000 neurons on each side.
And in humans?
Apparently, it's not that many more. And it's an interesting question. How many of these cells do you need in order to have a brain clock? And it's not that many. So 10,000 on each side in rats and humans, and maybe 10,000 or so in humans altogether.
Yeah. And in hamster also,
Yeah,
around 10,000.
Yeah.
This is amazing. Like, although the brain size changed almost
that's right.
100 or 1,000-fold even.
Right. So given that the brain size changes, but the size of this nucleus doesn't change anywhere near as much, it suggests that the output, the target of this brain clock is nearby. Because if the target were in your toe, it would have to get bigger in order to support anything that would go all the way to your toe.
Yeah.
So the target of the nucleus must be nearby.
Yeah. So the target means, in this case, parts of the brain that receive information, the first
Right.
hand, the first relay, the first person in the relay race.
That's
who are
right,
getting
right. That's
getting
right.
the information. And why studying the SCN has been such a lifelong passion for you? Can you tell us what is the importance of this SCN?
So I actually was still trying to figure. I had worked out how hormones in mammals, in the first animals were the hamsters. We worked out how hormones act on the brain, and that was pretty much directly out of my background. But I was living in New York, and I had moved there from Canada, from Montreal, and I found myself stuck in New York waiting for my husband to finish his PhD, because I finished faster than he did. He was doing theoretical physics, and that took longer. And I couldn't go anywhere, and it was the time when people were doing transplants to try to cure Parkinsonism. And
Really?
yeah.
Transplant, brain transplant to...
Adrenal tissue transplants. We had discovered that for the Parkinson problem was a decrease in dopamine, and so people were trying to find alternate ways of providing dopamine to the brain. And it was done in Sweden, it was done in Mexico, and it was an era people were trying to solve spinal cord transection with some sort of transplant. In any case, Eric Bittman was visiting New York. Michael Lehman was a postdoc in New York. Marie Gibson had been doing transplants in the GnRH system, and her transplants worked. So I guess I have to back up. Remember, I'm coming from a neuroendocrine background. Charlton in England had developed these mice that were lacking in GnRH. And there's a human disease, the Kalman syndrome, where the neurons that support the hormones, the GnRH hormones that support all of reproduction, get stuck. They don't migrate properly from the brain from the periphery, from the nose into the hypothalamus. They get stuck in the cribriform plate. And so Marie Gibson was working with Dorothy Krieger, and she transplanted GnRH neurons into the brain of animals who didn't have them in the hypothalamus. And she was able to restore function.
So these are gonadotropin releasing hormone neurons. And for our broad audience, can you tell us why these neurons are very important, and what does the human disease look like?
Okay, so the gonadotropin releasing hormone stimulates the release of hormones from the pituitary called luteinizing hormone and follicle stimulation hormone. And in males, they support testicular development and sperm production and all that. And in females, they support ovulation. So in Kalman syndrome, they don't have testicular development.
Yeah. And also for females, there is no luteinizing hormone.
Right. So these transplants of the GnRH neurons restored function. And so that was really exciting. The Parkinson people didn't have much success, but Marie Gibson's work was successful. And she had been an undergraduate student of mine. So this is a lesson. Always be kind to your undergraduate students and others. And I said, Marie, would you help us, teach us how to do transplants of the brain clock? And so we all hung out together and did this research, which at the time seemed a little crazy, because It's fine to say that this is a clock and it oscillates in a dish, but in order to prove that it works as a clock, you have to take my clock and then use my time, right?
Yeah.
So that would be real proof. Anyhow, then we did those transplants and they worked. And I thought, wow, this is really exciting. How could transplants work and where can we use them? And so that set me off on many years of following up on that story.
So the transplant was between two different animals?
Yeah.
Yeah.
In the end, the definitive experiment was to transplant the animal who had a fast clock. If I have a very fast clock and I take a transplant from you and you have a very slow clock, then if I'm using your slow clock, I will be late everywhere I go. And that was the idea. We had a donor animal that had a fast clock and a host animal that had a slow clock. And the recovered rhythm after the host's own clock was destroyed, the recovered rhythm was that of the donor. So it proved that the donor's clock that was transplanted into the host was the one that was controlling the behavior.
So what you're transplanting is this all or part of this suprachiasmatic nucleus. So you're taking this tiny part of the brain, which is almost, for most people, they may not even think that this is part of the brain.
Right.
If you squint your eye, you can see a tiny, like, it's even smaller than the tip of a pin.
Oh,
Almost.
well, no. So maybe, let's say,
Hair width of the-
500 microns by 350 microns.
Yeah. So it's less than 1 millimeter by 1 millimeter. And so that was a very exciting paper at that time, even till now, because I don't think any other, or maybe you can correct me, is there any other instance where you can take part of the brain of one animal, transplant to the other animal, and you can transfer all the information from it?
So, okay, so I wanted the GnRH system that I was describing to you, where you transplant these GnRH neurons, the neurons that support LH and FSH and all of reproduction, that works. So,
but that's a few neurons. That's not anatomically defined brain region, right?
No, because the GnRH neurons are a little bit scattered in the preoptic area, but it has to be those neurons. The fact that they're spread out a little bit isn't really...
Doesn't matter.
Isn't a story killer. It's still transplantation of specific neurons.
But this is a very great example where, something else, somebody else working on a very different problem, found a solution, and then you use that solution to address the very important question. And
Right. And when we first did the transplant, nobody believed us. It was really hard to convince people. We had to put all the records and all the anatomy up on the wall and show that every single animal with a good transplant recovered. This was also the only experiment I've ever done with no variation. Every single animal with a SCN in the transplant recovered behavior. So it was a bit shocking that it worked so easily, but subsequently many other people were able to do it. It's reduced to practice. It's once you find it, it was easy. So the next big question was, well, how could it be working? And why aren't people using it who study other systems like the Parkinsonism? Why doesn't that work? Because those didn't work, but our system worked like that. And so we tried to figure out how it's doing the restoration.
But when you're doing this transplant, there may be some animals which may be getting half of the SCN or 1/4 of the SCN. Was there any minimum size of the SCN that would make it work?
No, in the transplants, they were always at least the entire SCN. But we did do a different kind of experiment to ask, do you need half the SCN, 1/4 of the SCN? So in the old literature, it's said that you can lesion one side, because of course it's bilateral, and you still get rhythmicity. You can destroy 50% and you still get rhythmicity. And it was believed that it didn't matter which part you destroyed, you still got rhythmicity. But subsequently, when we understood how many different peptides are in the SCN and how they're organized locally, we were able to show that if you lesion the core area, we called these micro lesions, if animals that had the core lesioned lost rhythmicity, but if they had only the shell, they weren't rhythmic. So yeah, it is specialized within the SCN, but we didn't show that in transplant studies.
So now you're triggering a couple of questions.
Okay.
One is going back to the animal study. Suppose the animal with a faster clock, you took out only half of the SCN.
We didn't do that.
No, suppose.
Suppose.
And then you transplanted the other half with a slow clock.
Uh-huh.
What would happen to that animal?
So I could tell you things that we did that were like that, not exactly that. We had a host animal who had a slow clock and donor animal who had a fast clock. We left the host in place and gave him a second one. Okay.
Yeah.
So now he has his own and the transplanted one, and we had his own clock in a light-dark cycle, and the donor in a dark-light cycle.
Yeah.
And we asked, okay, who wins here, the host or the donor? And it was always the host who won. So the donor tissue came into sync with the host.
Oh, okay. So that explains some of the
something. Yeah.
But then going back to this observation that you mentioned, that although the clock is around 10,000 neurons, if you take out the middle part, suppose say the core, then that's what matters the most. So then the question is, well, going back to now two questions. Now going back to your hormone effect on the clock. Although you came from the dove taking care of their babies, in humans, as people go through puberty, does sex hormone affect the circadian clock?
Well, Till Roneberg has the data that seems the most beautiful to me, where he looks at chronotype. What time do you go to sleep? Okay. And he shows that females go to sleep earlier than males until about late adolescence or early 20s, and males go to sleep later than females. And that this sex differences decline slowly with age until adrenarche, menarche, and where there are no more sex differences. So that suggests that the pubertal hormones impact timing of sleep-wake cycles.
But you can also take the SCN, and you and many other people do this experiment. You put it in a dish, the SCN would still cycle.
Yeah.
And you can measure the rhythm.
Yeah.
In that juice, that is it.
Yeah.
If you are sex hormone,
yeah.
Do you see any difference?
Yeah. Well, I've never done experiments like that. And one of these things that stopped me from doing it is the medium that people use to culture tissue like SCN has a lot of steroids in it.
Oh.
And so you would have to work out how to do a medium that had no steroids and still supported rhythmicity, and then start figuring out what concentrations of steroids to add. So I don't know. Maybe someone did it, but I don't know the answer.
Yeah, means I was thinking.
And you have to watch out for the medium.
Yeah, but that makes sense, because a lot of the mediums do have a huge amount of steroids. And
and steroids get metabolized into the what's called sex steroids.
Yeah, sex steroids.
Yeah.
But then the second question relating to this, the function of the core of the SCN. As people get older and their neurodegenerative disease, like Alzheimer's disease, do SCN neurons get affected? Do they die? Just like the other parts of the brain die? And if so, does that explain why in many neurodegenerative diseases, people experience random sleep-wake cycle?
Of course, that's a really big question, and a lot of people are trying to understand that. I've never seen any evidence that the SCN itself dies, but there's evidence that the rhythms that are orchestrated by the SCN become disrupted. And we need to know why they become disrupted. So something about SCN control of rhythms in the rest of the brain and body has changed. My own favorite guess, this is guess, not solid data, I've become very interested in circadian effects, SCN effects on the vasculature and on the CSF, cerebrospinal fluid, and on interstitial fluid. So I don't know if you've been following that work, but what we have shown is that the SCN communicates with these nuclei that lie all around the cerebrospinal fluid in the third ventricle. And they all communicate with each other. And we didn't cover it yet, but my work with the transplants shows that even if the transplant is put into a polymer membrane, like a bouquet garni, when you make soup or stew, it still works. So it means that the SCN transplant is working by sending out a diffusible signal. So the SCN sits right by the ventricle, right at the base of the brain, and it sends out a diffusible signal that probably reaches these leaky brain areas. So my favorite thinking these days is that the interstitial fluids in these leaky brain areas are part of how the SCN brain clock signals. And very exciting work is being done by Maiden Nadergaard at Rochester. She's given a name to these spaces around blood vessels. She's called them glymphatic spaces because these systems seem to be like the lymph glands of the body, but they're in the brain. So in the brain, they're called glymphatic. And I think that circadian signals regulate the activity of the glymphatic system. And that system is important in clearing waste from the brain, and that's what seems to happen during sleep.
So,
as you get neurovascular disease, that falls apart a bit. That was a very packed answer. Maybe you could deconstruct it a little bit.
Yeah, we can break it down to a few things. One is most people, when they think of neurons, because suprachiasmatic nucleus, a lot of neurons, 10,000 neurons, we always think that the nerve cells, they go touch each other and send the information through what we call synapse.
Yeah.
But at the same time, some neurons can secrete stuff like hormones, peptide hormones or other hormones, and that can go through the bloodstream. Typically, hormones go through bloodstream and affect their messengers. They go to the other parts of the body or brain and unlock something or send the signal. And what you're saying is, since you could tie up the SCN in a semi-permeable bag and drop it inside the center of around the base of the brain, where the fluid is sloshing around and taking all this and circulating through our spine. So that means that the SCN neurons did not have to send their hands to touch another neuron, but they just secreted something, magic molecule, hormone-like molecule into this fluid, and then that fluid transmitted all the information. Because the SCN regulates, say, sleep-wake cycle, eating, fasting cycle, osmolarity, many other things. And all these brain regions are kind of lining up around this third ventricle or the sloshing around CSF.
Yep.
Yeah. And but at the same time, it's exciting that so that means what you're saying, although in transplant experiment, you are dropping the SCN in a bag, but in real SCN, which is not touched, what you are suggesting is SCN is in constant communication with this lymphatic system. So that means SCNs there is no blood-brain barrier between the blood and the SCN?
Well, what we have shown is that the SCN sends a special vessel called a portal vessel to the leaky blood, the leaky nucleus called the organum vasculosum lamina terminalis. Terrible name for a nucleus, but anyhow, let me call it the OVLT. So the SCN has special blood vessels that go very, very locally to this local target. And that's a very leaky part of the brain. It has really high density vasculature. So we showed that system in mice and in rats. And at this meeting, June Yao showed that it also happens in humans. And for sure, it's ubiquitous through all mammals, all vertebrates. These diffusible systems are evolutionarily much older than neurons. So, yes.
But it's only, so there are very few, very rare examples of brain regions using this specialized way to send signal. Is that right? Are there other examples?
The first was discovered, the first was discovered in 1930. The hypothalamic pituitary axis, that has these specialized portal vessels. The second was the one I just described 50 years later, called the SCN OVLT portal system. And once we saw number 2, I said, oh my God, I don't really want to do this research, but I should find out if there's more of them, because maybe it's a common way of communicating in the brain. And so we looked at other nuclei that line the ventricles, and each of the sensory circumventricular organ has such a system of communicating by the blood system locally. And they each have access to the cerebrospinal fluid. So the metaphor I'm using in my head to keep track of this is, well, it's like roads and rivers and oceans. So the neurons are like roads, and these special portal systems are like rivers, and the CSF is like an ocean. And, you know, I live in Manhattan, and there's the Hudson River. Manhattan's an island. And when the tide rises, the ocean runs into the river. And when the tide falls, the river runs into the ocean. And in the CSF, there are daily changes in CSF pressure.
Yeah.
And they're in the antiphase to blood pressure changes in the periphery. So I'm imagining that these systems regulate circadian rhythms widely through those CSF and interstitial fluids.
Yeah. So now
it's only logical.
No,
it doesn't mean it works that way, but it's logical.
No, this is really interesting that what you're saying, this kind of specialized way of SCN sending information to other parts of the nearby, to their neighbors, may be more widespread than previously thought.
Yeah.
And that's a big discovery in neuroscience, because it changes our thinking of all neurons communicating through roads to neurons maybe using the waterways to communicate.
Right.
Yeah.
That's right.
And that's like in 1930, and then after 50 years or so, you discovered, and now it seems to be rediscovered, and maybe it will change the way we think about how the hypothalamus, or part of our brain, communicates with the rest of the body.
Yeah, so neurovascular disease and changes in the vascular system are becoming targets of interest of researchers, because it appears as though neurovascular disease underlies many of these cognitive-related diseases like Alzheimer's.
Yeah.
Yeah.
So just like, so that brings up another question, like there is, when we are talking about neurovascular system, that's blood vessels inside the brain. And we know that when the blood pressure goes up, or there is a clot, for example, then that's a clear example of how the vascular system affects the brain. People get a stroke.
Yeah.
Yeah. So in that way, as you said, the blood pressure and the CSF pressure goes hand in hand. Are there cases where the CSF pressure is too high, just like the blood pressure goes up high?
You know, it's really hard to measure CSF pressure, because as soon as you start measuring it, you change it.
Yeah.
So it's not something that's universally agreed upon. It's a big problem for people who have hydrocephalus, for example, but we don't have ways yet of monitoring under normal, non-invasive methods, pressure changes and flow. We don't have that yet.
Yeah.
You can measure, you can make a little window and look at the cortex, but any way you inject a tracer, you can't do it in vivo. So we don't know what happens in real life.
Yeah, so that's the biggest mystery, because
it's the biggest mystery.
Brain is an organ. If you open to look at it, you essentially change it.
It's a problem. Especially for pressure and flow. Very hard to measure.
But then, okay, so coming back to the SCN, and you have worked on it most of your life, what are the parts of the brains that receive information from the SCN, and what do those brain regions do to the rest of the body? Because when we call it the master circadian clock, it's sending that timing information to coordinate sleep-wake, eating, fasting, bowel movement, many other things, almost everything in our body.
Yeah. Well, from the first time of its discovery in 1972, people have done track tracing of the roads. And so that's a big established literature. And people have tracked not only the first synapse, but also second and third. Larry Morin has a big review paper showing that the SCN communicates widely through the brain if you look at second and third relay stations. And what I'm saying is, on top of that, by these rivers, SCN can communicate through the whole brain, because every blood vessel has a space outside of it. And in 18-something, sorry, I can't remember the year, that space was called the Virchow-Robin space, okay? So sometimes when you try to read the literature, it's so confusing because you don't know what they're talking about. But that space outside of blood vessels was called Virchow-Robin space. It's also called perivascular space. And in the new hot studies looking at sleep function, it's called glymphatic spaces. But all of them are the spaces around blood vessels. And what our portal system work suggests is that this is a way that the SCN can relay the signal to a leaky blood area, a leaky brain area, and from there it can go to the whole brain. So you asked me, what is the target? Well, everything is the target.
So in that way, then SCN can actually connect or influence even learning and memory, which is not in the hypothalamus. The brain centers are away from there. So you have done some work on...
No, I never did learning and memory. But I should add, if you look at the brain vasculature, the hypothalamus, the oldest part of the brain, is incredibly densely vascularized. It's like, if you look at... I know you live on the West Coast and I live on the East Coast. If you look at the rivers of the United States and the flow of rivers, the East Coast has a lot of water. The West Coast has no water.
Very little.
The hypothalamus has a lot of blood. The cortex, not quite as much. So...
I never thought of that way. And actually it makes sense, because when we do immunostaining or even the electron microscopy serial block test EM, we do see a lot of blood vessels in our images, but not from other labs where I'm like, what are the blood vessels?
Where's the blood vessels, right?
And another thing is SCN is also very densely packed. The number of nuclei per unit area, like it's almost like Manhattan. It's not like,
Right.
It's not like this suburb.
And we heard at this meeting that it's the capillary density of the, well, we actually did the anatomical study on the capillary density, but in humans, who was speaking about humans, Jun Yao again,
Jun.
He described this incredible density of blood vessels in the human SCN and OVLT. It was really impressive.
So it's very different from the rest of the brain.
Well, it might be,
like the top of the brain.
yeah, might be similar for other hypothalamic structures. I don't know.
The top of the brain
Yeah,
is
yeah.
very different from the bottom of the brain. And then you also, means, the thing is, recently I read this review that you wrote, because you said in 1972, SCN was formally linked to circadian rhythm, and it's almost 50 years that happened in 2022. And you actually had a beautiful review, 50 years of SCN.
Oh, yes, that was fun.
And I think all the neuroscientists who want to learn a little bit more about SCN, that's one of the best comprehensive review, very well written for a lot of people who are not even in circadian rhythm field. They could follow it.
We had brilliant colleagues, of course.
Yeah, thank you. That was it. What else excites you in circadian rhythm, SCN? Because now circadian rhythm is connected to so many different things. The immune system and neuroimmune function is now a big thing. And almost everything in biology is exciting. And everything is connected to timing.
Yeah. So what excites me is the potential for this research to be used to help us live better lives. And the part of it that really impresses me is the chronotherapy, optimizing the use of drugs to have the least adverse effects and the most beneficial effects, so that when drugs are given at a correct circadian time, they're more effective. And I think the most exciting thing I heard at this meeting was somebody who was studying cancer cells. And I'm sorry, I couldn't, it was a lot of data, but the principle he laid out was, if you give treatment he compared treatment at time 0 and time 12, or maybe it was 6 and 18, I don't remember, but 12 hours apart. And the drug he was using lasted about 2 weeks. But what he showed was, depending on which drug you gave first, drug number 1 or drug number 2, you had a completely different effect. So if you started with drug 1 and then followed drug 2, that was a different effect than if you gave drug 2 followed by drug 1. And he gave these 12 hours apart, but that first drug effect lasted for weeks. And he tried to figure out how that worked. And what he showed was that the cancer cells are either in the tumor or dispersed out of the tumor. And depending on where you gave that first injection, you trapped them either in the tumor or out of the tumor. So it didn't matter what you did second, because the first drug actually changed the state of the cancer cell.
Yeah.
Did you see that, Tom?
Yeah.
Oh, so you probably could explain it better than I just did.
That's actually...
It just blew my mind.
Yeah, so these are some of the really exciting things that are happening, because we always thought that the drugs that clear out within an hour or two, maybe time of administration has an impact. But then some of the new therapies, like the immunotherapy, which, cancer patients are getting it only once in every few months.
Every 3 weeks, yeah.
Yeah. Or even, for example, in extension, maybe we should think about all the GLP-1 receptor agonists, all the incretin, the weight loss drugs that people are getting once a week or once in 10 days injection, whether getting the drug in the morning or evening would have an effect.
Yeah.
Because the first hit is almost like a resetting cue. It's like you are startled. It's like, I think it's very similar to a guest coming to your house. If the guest comes at 10 o'clock in the morning after you have finished your breakfast and in a good mood, then the rest of the visit will go very well. But if the guest comes at 1 o'clock at night, then maybe,
But the power of that effect was so strong.
Yeah.
Very impressive.
Yeah, actually, he presented this data we saw maybe last year or something, because we are doing similar studies in mice, and we thought, okay, we should not reinvent the wheel. We should take whatever he has done and he already figured out what optimum timing should be. So we are treating the mouse with cancer with this timing.
Yeah.
And we are seeing some exciting results. So going back to more exciting stuff that you think this field will give to the community. One is treatment. But the challenge is most drug companies are saying, well, we already put the adverse effect, and physicians are also accepting that there is adverse effect. They're warning the patients that there will be some adverse effects. So do you think that's the most impactful stuff, or is there something else?
So the drug stuff hits one community, right?
Yeah.
But the other impact is lifestyle, the kind of work that you've been doing, convincing people that making small changes in their routines on eating or exercise or exposure to light, if we could convince people that that works, that would be a big contribution. So the trick is, how could you possibly get compliance, right?
Yeah.
Yeah. So I think the wearables must at least give people information what they're doing. I'm actually curious to hear your opinion on whether we're going to have impact with those lifestyle suggestions.
Yeah. So, coming back to your point about light, and let's revisit that, because we talked about how SCN transmits the information out through rivers and highways. But the light information from the retina, they come largely through highways or the roads to reach the SCN neurons.
I think so.
Yeah, because so far we haven't seen any other way.
No.
So this is also important. Although the SCN is the master timekeeper, it also has to synchronize to the outside world through light, primarily coming through the eye, hitting the SCN. And over the last, 2 to 3 decades, there are new studies showing that food timing can also change some of the clocks, but food timing does not change the SCN. So that means we have to be careful about when we eat and when we see light. So I think this is where you are bringing up one important point, that we built this anthropogenic world, man-made world, without paying attention to when we get light, when we eat. And now that we have 60% of the population overweight or obese, and nearly 50% have hypertension, 50% have pre-diabetes or diabetes, and people might think that, well, pop a pill and most of the diseases will be gone. But it's not that easy. So then the question, what you are pointing out is what I am hopeful about, whether this lifestyle intervention works. I put it this way. So for example, when a pre-diabetic person in the US becomes diabetic, type 2 diabetic, then the average annual healthcare cost for that person goes up by $9,000 to $10,000 a year. And if this person is becoming diabetic at the age of 50, then for the next 30 years, we are spending $10,000 a year on an average for 30 years.
Yep.
And we have 30 million people, or 40 million people with type 2 diabetes. So if we can implement, and there are nearly 100 million people who are pre-diabetic. If out of 100 million people, only 1 million people, my hope is only 1%. Let's start from there. Only 1 million people, say, adopt this, pay attention to lifestyle intervention. And in fact, we did this experiment in some small cohort. If they do time-restricted eating, that was the only intervention we did. But if they pay attention to light, that may be even better. And by doing it just for 1 year, if they delay the onset of diabetes, I'm not saying that they will completely prevent diabetes, by just 1 year. So that's $10,000 for 1 million people. So that's $10 billion. So even that will have huge, huge impact. That's almost 100x return on investment into all circadian rhythm research done over the last 20 years.
So to whom would you make this argument, though? And then how would you implement it? Because you need all these individuals to act, right?
So I think that's already happening, because at least there are now 3 or 4 apps that have more than 1 million downloads, which are recommending time-restricted eating, sleep, and circadian rhythm. So that means there are already, say, 4 or 5 million people who are getting the message and are also paying to get them monitored. And these are some of the paid apps. So that's one way. And then the second way, I think we have to also, as a society, European Society of Biological Rhythm or US SRBR, we also have to come up with what is the impact of this research. We work with honest taxpayers' money. And they also have to understand that your interest in figuring out how the male and female dogs take turn to raise their babies, actually led to the major discoveries about circadian rhythm. And that's going to impact health. And that investment is coming back so that everybody is going to reap benefits. And this is one exciting thing, is there is no cost involved.
Yeah.
People can implement it in their own life.
Yeah. So one of the thoughts I had was that, of course, we want to get it into medical practice as well. And one of the thoughts I had is, I'm always impressed with when I give a lecture to the undergraduates. They love these stories, and they are very interested in losing weight or feeling better or not being depressed. And so a significant number of the undergraduates try this out. And so we can give them little Fitbits or something and have them monitor and share this data. And for my cohort, they're neuroscience students. And they all go on to medical schools, like 90%. So by tackling the young people, undergrads, which is I have access to, but maybe high school students as well, they will carry this forward. And in the long run, we can have a broader impact both on lifestyle for individuals and in medical practice, where it's very hard to introduce new topics because they have such a heavy workload already.
No, that's very exciting. That's a great idea. Maybe we should talk about it a little bit more offline to figure out how to come up with a very structured program for undergrads in Columbia.
Yeah.
And once we set it up, then we can set it to a UC system and many other systems.
Oh, that would be great. I would love to work on that.
Yeah, let's do that. But at the same time, you brought up another important issue, undergrads, and then they becoming doctors and physicians and all that stuff. And when I talk to people, young people also come up with this thing that, well, the time between when they're 20 and 40, there are a lot of life changes they're going through. They have to find a new job, they have to start a new family maybe, they're buying a new house. At the same time, many of them have to care for their siblings or the elderly. Any advice?
So life has its challenges and quiet moments. And maybe you can coast along with the calm life for a long time, but eventually you're going to have challenges and alternating with calmer moments. And what I have found is that it's great to be able to work on something outside of your home, to have something that is interesting and occupies your mind. And so being able to do research is like having lifelong puzzles. It's the most wonderful lifestyle. So maybe not everybody can be a professor doing research. I mean, that would be a ridiculous aim. But I think everybody can have some passion, some interest outside of their ordinary daily life that they escape to when you hit these moments that are difficult. So I just encourage people to have a passion, a love. And if it's research, great.
Yeah.
Go for it. Even if you're challenged finding a job, just keep trying. Do what you can and then find something you love.
That's very true, because when people ask me, I always say that you should have your work, family / relationship, and a hobby or passion.
A passion.
Yeah, and sometimes, like, we have been lucky that our work is our passion, so we don't have to look for. But you also have passion because you are a great photographer, or you try to be, and I also try to be.
Love photography.
Yes, so we both love photography. But this is a key thing, and also this is what I appreciate in you is most people think that scientists are very dry and they are always in the lab and they're trying to write their next grant or manuscript, or they're critiquing, writing reviews, criticizing. But actually, we are not that bad. We are very warm people.
Yeah, my sister says, you're always looking at the trees, never the forest. So even the family attacks at that level.
Okay, here's the hardest question. Looking back at your career over the last X number of years, are there 1, 2, or 3 key discoveries that you're really proud of, or that gave you that jolt saying, yes?
There are 2 that it shocked me that the transplants worked so easily. It was almost unbelievable. And then it shocked me that it involved a diffusible signal. So those were, and at the time we did that work, some argued that, I don't know if you remember, but there was a Japanese group that showed that if you cut along the SCN, just made a knife cut, that you lost rhythms everywhere. So that seemed to contradict what we found. But following up on that, at this meeting, I heard really beautiful research from a young person named Shota Miyazaki. I hope I have it right. And he created that same island, he recreated that original work and showed that circadian locomotor rhythms survive following the island. And what's even more impressive is he's following up on that to figure out what the signal is that comes out of the SCN, and he thinks it's BDNF. And they have very nice data for that. And now he's looking for the target of that BDNF. So it's a follow-up of years of research that we can now tackle. So there's great joy in seeing it, your original finding rediscovered in a new context and moved forward in a way that we couldn't do it at that years ago. So that's one big thing. And now the discovery of these portal systems that we've already talked about really blows my mind. It means we have to rethink how the brain communication systems work. And for sure, I can't do the necessary follow-up work. It's a huge endeavor. I hope that gets picked up, and I hope I get to see it. So those 2 findings.
But those 2 are also very high-risk project.
Yes.
So, but in between, you co-starred with many other publications. So, any advice for young researchers?
Yes. I think for me, what I've always done is to have something that's high risk and something that I know I'm going to get an answer. I always have an answerable set of experiments going and something where I have no idea whether it's going to work, not work, but might be really important. And most of the really important ones don't really work out. They're high risk, and they prove they're risky because they don't work. But having both going keeps your attention and excitement level up. So I always have 2 types going. I think it's a good way to organize your work so that you don't go home and get depressed because, oh God, everything's failing. I mean, research is slow and you have to be patient and you have to try things in many different ways. And you just want to have little successes as you move forward.
That's true. I mean, I say that in every 5 to 10 years, if you have an aha moment, you're a very successful scientist. But between those aha moments, you have to keep the lab going. You have to train students. So you should have this very confident experiment, which are about most likely to succeed.
Yeah.
Yeah.
Yeah, for sure.
Yeah. So it's really exciting, and it's very, for me, it's an honor and privilege to sit down with you, because almost every circadian rhythm researcher, when they start on this field, they read those very original SCN transplant papers. And that established essentially that the SCN is the master circadian clock. And also when I worked on melanopsin blue light sensing ganglion cells, the challenge was very simple and straightforward. Do these cells connect to the SCN? If they don't connect, forget it.
Right, right. And we should also give credit to the follow-up studies from the original transplant papers. The transplant work that we did with Marie Gibson teaching us showed us how to do it and that it worked. But the real knockdown that this is for sure the case comes from when Martin Ralph had discovered these mutant hamsters and that we could transplant animals with different periods. So again, a great finding followed by follow-up is an even greater finding. And so we're always building on each other's work.
Yeah, that's true. We are always building on each other's work. And so that's why sometimes we have to give credit, and we should be very humble.
We should be humble.
Yeah.
We disappear, but the research carries on.
That's very true. Thank you, Rae. It has been a great pleasure speaking with you, and hopefully you all will enjoy this. And you actually got to listen from one of the leaders and legend in circadian biology. As Rae pointed out, the work will live on. Thank you.
Thank you, Sachin.